Okay, I think we are on now. So, so our last class, like I said, we went through mitosis, and at the end of the whole class, we were able to derive that somatic cell, which is body cell, make use of mitosis, growth in human being, or growth in that's in multicellular organism, and even growth in what's it called, reproduction in some is in some unicellular organism like amoeba. They undergo asexual, that's asexual reproduction. So they make use of mitosis. Um, a parent a parent cell matured and develop one divide into two to give rise to daughter cells, which also have the same number of chromosomes as the parent, which is the diploid number of chromosomes. So we are coming back to meiosis here. And basically, I just want you to know the difference between mitosis and meiosis. Basically, meiosis actually help in the formation of gametes. That's sexual reproduction. It helps in sexual reproduction. So gametes that are used during sexual reproduction, meiosis really help us to produce it. If you look at the diagrams, the two diagrams I have here on this page, you will see the sperm cell from a mature male organism, and the egg cell from a mature female organism. And we'll still go more into that. When we get to sexual reproduction, we'll talk more about that. But basically, we just want you to know that meiosis help in the production of these sex gametes. Are you getting me? That's what I want you to pick up from here. And since you are online, like looking at this now, it's good if you begin to know the diagram of a sperm cell and egg cell, and just know everything, the acrosomes, what it is used for, the nucleus in it, how many number of chromosomes you have in the nucleus, the collar, know the function, know what it's used for, the flagellum, the nucleolus, the this and that. So from there, you'll be able to um, start developing yourself. So by the time you're able to go deeply into reproduction, so it will be very, very easy for you. So let's go straight to the point for today. So the production of offspring by sexual reproduction includes the fusion of two gametes. Now, when you want to form another organism, we make use of sexual reproduction. That's why I decided to highlight it, so you'll be able to know all this necessary information that you need to understand under them. So two gametes come together. From the male, we have the sperm cell. From the female, we have the egg cell. So during reproduction, after copulation, the sperm cell fertilizes the egg cell, and you give birth to a zygote. That's a new offspring begin to develop. If you remember, under this very topic, what's this topic called? That should be the second topic after mitosis, embryology. Yeah, under embryology, I was able to explain how this happened. When sperm cell fertilizes egg cell, what happens next, and all those stuff. So you can go back to that topic to understand it also. So each of these gametes has 23 number of chromosomes, which is also called haploid set of chromosome. Haploid chromosomes. Haploid means half of diploid. Diploid, D-I and ploid. It means two. Haploid means one. So two sets of haploid give you diploid. Two sets of haploid give you diploid, meaning 23 plus 23 will give you what? 46. That is in human being, for example. I'm using that because not all organisms have the same. All organisms have different chromosomal number. When we get there, we'll talk more about that. So let's go straight to where we are going to here. Now gametes are formed from specialized diploid cells. I've mentioned diploid cells under mitosis. Specialized diploid cells give rise to gametes, meaning the diploid cell go through reduction and give rise to what? Gametes, which is half of them. Now, it reduces the chromosomal number or the chromosome number by half, result in the production of haploid daughter cells. So, half of the diploid cell will give your what? Haploid cell. So, there is a what? There is a reduction. Now, meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organisms, whereas fertilization restores the diploid phase. Now, meiosis, look at your diploid cell, which is from the somatic cell, the body cell, the specialized ones. It will make sure it reduces it into haploid phase. It will reduce it into haploid, and fertilization will restore it back. What am I trying to say? The 46 number of chromosome from the somatic cell will be reduced to 23 in male, and the 23 will be stored inside our sex gamete, e.g., the sperm cell. The same thing will happen in the what? In the egg cell. So during fertilization, when your sperm cell now fertilizes your egg cell, 23 plus 23 will now come back together to now give what? 46. And that is why after sperm cell fertilizes egg cell, you have a newly developing embryo that will now begin to develop to give rise to what? To a newborn baby after a period of time. So what we are trying to say is meiosis will reduce your diploid cell into haploid cell. And fertilization will now bring two haploids together to now give what? Diploid, which will be a new offspring or organism. Now, what are the key features in meiosis? What are the key features? What are those things we are trying to look out for under this topic? Now, meiosis involves two sequential cycles of nuclear and cell division. Now, if you remember very well, we talk about cytokinesis and we talk about karyokinesis. The same thing is what we are trying to refer to here again. Meiosis now it involves two sequential cycles. It's sequential, one after the other, of nuclear division, meaning there will be nuclear division first, followed by what? The cell division. Now, this phase, compared to mitosis, in meiosis, we have meiosis 1 and we have meiosis 2. But mitosis is just one phase, just mitosis. That's just it. But in meiosis now, we are going to have meiosis 1 and meiosis 2. Now, meiosis 1 is initiated after the parental chromosomes have replicated to produce identical sister chromosome at the S phase. You remember very well, we explained the synthesis phase, the G0 phase, the G1 phase, and the G2 phase. Yeah, I explained it under mitosis. It's the reason why I decided to separate these two classes so that you would have gone through the mitosis and understand it very well before you now get into meiosis. And as the same way I'm explaining here, as you are watching this and looking at this, the best thing is get your jotting material and jot it, summarize everything that you need so that you'll be able to digest it properly. Now, meiosis 1 is initiated after the parental chromosome have replicated. Now, the parental chromosome, the organism is fully developed now. So we have parental chromosomes, meaning for the adults that want to go into reproduction now. It has replicated to now produce identical sister chromatids at the S phase. Don't forget, S phase is the synthesis phase. And at this phase, there will be production of what? Chromatids. If you remember what I explained under mitosis, chromatids will be what? Will be produced from chromatin material. Chromatids will be produced from chromatin material, and chromatids now come together. They attach themselves at the centromere to now give the what? the chromosome. I think you can still remember all those explanations. Now, meiosis involves pairing of homologous chromosomes. Remember that homologous chromosomes. Homologous chromosomes are chromosomes that have the same size, shape. They look alike. They're identical. Are you getting me? So those are the ones that pair. If they are not homologous, they will not pair. So pairing of homologous chromosomes and recombination between them. Homologous chromosomes, they have the same length, same height. They pair together, they come together and they pair. And there will be what we call what? The recombination between them. Now, all those processes, I will explain it in meiosis 1. Most of them occurs in meiosis 1. Almost every one of them occurs in meiosis 1. So as we are going through that, you'll be able to see what recombination is and how homologous chromosome looks like. Now, 4 haploid cells are formed at the end of what? Meiosis 2. 4 haploid cells are formed at the end of what? Meiosis 2. So which means instead of in mitosis, where you have 2 diploid cells being formed, here under meiosis, it is going to be what? 4 haploid cells that will be produced. So let's continue. We'll understand that as we move. Now, meiotic events can be grouped under the following phases: meiosis 1, meiosis 2. Now, when we talk about meiotic is the same thing as meiosis. Are you getting me? So meiotic, mitotic, meiotic. Still referring to mitosis, meiotic. Sorry. Mitosis, mitotic, meiotic, meiosis. So it's just to talk about the event. So the event now, they make use of tick and other stuff. So in meiosis 1, we have prophase 1, metaphase 2, anaphase 1, and what? Telophase 2. 1 rather. Everything 1, 1, 1, 1. In meiosis 2, we have prophase 2, metaphase 2, anaphase 2, and what? Telophase 2. Now, let's take them one after the other so we'll be able to understand them better. Now, in meiosis 1, we say prophase of the first meiotic division is typically longer. Now, longer and more complex when compared to the prophase of mitosis. In the prophase of mitosis, it is very short. It doesn't take time at all. Yeah, it doesn't take time at all. In short, the prophase of mitosis, please, I want you to understand what I'm trying to say clearly. The prophase of mitosis is the same, almost the same, as the prophase of meiosis 2. But meiosis 1 is longer than the prophase of mitosis. I think you understand what I'm trying to say now. So we are trying to say it's a long process because a lot of things will go on in prophase 1 of meiosis 1. Okay, so now let's look at what we have here. Under prophase 1 of meiosis 1, we have a kind of subdivision. It's subdivided like stages under prophase 1 of meiosis 1. Now, under prophase 1 of meiosis 1, it is subdivided into leptotin, zygotin, pachytin, diplotin, and diakinesis. So I gave it a formula, which is what is right in front of it here, which is LZPD2, meaning 2D. We have 2 of D there. We have L, we have Z, and we have P. So you can rearrange and put it in any form you feel you will quickly remember that. Now, let's start from the first one, leptotin. So in leptotin, chromosomes become gradually visible. Okay. this is the diagram here, as we can see here. Remember that in the synthesis phase, G1, G0, G1 phase, if you remember the explanation under the preface of mitosis, if you remember that explanation, we said there is a resting phase, which we assume to be the interphase, that it is a resting phase. But interphase is now further divided into G0, G1, synthesis, and G2 phase. Yes. Now, what we are trying to see now here again now is the same thing that happened inside the interphase. Now, in the interphase, there has been synthesis, everything has been formed. Are you getting me? But it cannot be seen. It's more in form of chromatin material and what's it called? And chromatins. Yeah, chromatins. That is how they look like. But in leptotin is the first phase here now of prophase 1 under meiosis 1. So at this very stage now, you will now see the chromosomes, which are the chromatin material in interphase, which you cannot see in interphase. You will now see it becoming gradually visible under leptotin. And next, there will be compaction of chromosomes in the same leptotin. Chromosome will now begin to come together. How do chromosomes form? Two chromatids come together, attach at the center, which is the centromere, to form the what? The chromosome. So that is the same thing that is happening here. If you notice this, now it's becoming what? Visible. You can see it visible. Chromosome condenses, so you can see it clearly. It's been formed already. Now, after this has happened now, chromosome condenses and you can see it. It is now visible. And you will also notice that what's it called? The membrane surrounding it is becoming what? Is disappearing gradually. So that is at the leptotin. Now, after leptotin, then we now go to the next one, which is what? Zygotin. Zygotin. Now, in zygotin, the chromosomes start pairing together. You now begin to see pairing. Now, if you look at the diagram here, we call this bivalent forming, meaning two chromosomes coming together side by side, as you can see, side by side. So you have 1, 2, 3, 4. 2 for this, 2 for this. So we call it what? Bivalent. Two coming together beside each other. Now, where they now attach, they will attach to themselves and there will be a kind of connection between them, which is a complex formed between them, which is called the synaptomenal complex. Are you getting that? Now, it begins to form in between them, meaning they will be what? Attaching. So we call it what? Synapses. It is a complex, but the main thing here, which is being formed, is called what? Synapses. So let's continue. Now, chromosomes start pairing together, and this process of association is called what? Synapses. Each pair chromosomes are called homologous chromosomes. Like I told you, they will have the same size, height, and everything. Chromosomes synapses is accompanied by the formation of complex structure called the synaptonemal complex. We've talked about that too here. The complex formed by a pair of synapse homologous chromosome is called a bivalent or a tetrad, meaning 1, 2, 3, 4, tetrad, or bivalent, 2 year, 2 year, 2 year, 2 year, valent, valent, two of them coming together, or tetrad, anyone that's easier for you. However, these are more clearly visible at the next stage. So this process, you might not see it very well. It might not be very clear under the zygote, but when you get to the next stage, it becomes very clearer because you can see the processes taking place clearly. Now, the first two stages of prophase 1 are relatively short-lived compared to the next stage, which is what? Pachythene. Now, at the pachythene, It's a long thing. Now, look at the first diagram here. As you can see here, you can now see that the homologous chromosome have lie side by side. The complex has been formed in between them, and you begin to see synapses, right? Synapses in form of what? You can see crossing over too. So that's why they said it's a bit longer, because a lot of things happen at the Parkinson. Now, a bivalent has formed and crossing over has occurred. So let's just go through this so you'll be able to understand what I just finished explaining there. Now, bivalent chromosomes, remember bivalent, how we have bivalent, or we call it tetrad, okay, clearly appears as tetrad. 1, 2, 3, 4. They've now come together, tetrad. Now, crossing over occurs between non-sister chromatids. What do you mean by non-sister chromatids? That is chromatids from two different chromosomes. We have sister chromatids. Sister chromatids are the chromatids on the same chromosome. But non-sister chromatids are from two different chromosomes. Chromosome 1 crosses with chromosome the chromatid of chromosome 1 crosses with the chromatid of chromosome 2. That is non-sister chromatids of homologous chromosome. Homologous chromosome. This is one chromosome, this is another chromosome. They are homologous chromosome because they have the same height, length, and everything, but they are two different chromosomes, okay? Crossing over is the exchange of genetic material between two homologous chromosomes. I want you to take note of that. That is a definition that can be asked, short answer question. What is crossing over? Crossing over is what? You are exchanging genetic material between two homologous chromosomes. And in short, this is one of the reasons why you and your younger brother, your younger sister, why you are different in look, facial look. Because at this very stage, a lot of crossing over is occurring, a lot of exchange in between the chromosomes that will now form gametes. That will now form gametes to produce a new offspring or new organism. So the reason why you and your younger ones are a bit different, somehow you'll be different. You cannot look 100% alike because they have been crossing over and there has been different genetic exchange, okay, which is at this stage. Don't forget Parkinson. Now, enzymes involved in crossing over is called recombinase. Now, recombinase in what sense? We look at the word recombinase. They are recombining. Are you getting me? So the enzyme that you need for recombination is called what? Recombinase. Crossing over leads to recombination of genetic material of the two chromosomes. Okay? Recombination between homologous chromosomes is completed by the end of Parkinson. So at the end of Parkinson, recombination has what? Occurred due to what? Crossing over. Okay. So we go to the next one, which is the diplotin. Now, if you look at the diagram, before we go to what we have here, if you look at the diagram, you will notice that what? At this stage, everything that needed to happen has happened, right? And they will now begin to what? To separate. Are you getting me? The synaptonemal complex will now begin to what? Complex will dissolve or dissociate. Are you getting me? Therefore, you now see the homologous chromosomes separating from each other and stand on their own. So, dissolution of the synaptonemal complex and the tendency of the recombined homologous chromosome of the bivalent to separate from each other, except at the site of what? Crossing over. Now, the site of crossing over, don't forget, this site of crossing over, why is crossing over? Because we are trying to what? To exchange genetic material. So two different chromosomes now are trying to exchange genetic material so that new offspring with different characteristics and so on and so forth will be produced. So that brother, firstborn will not look like secondborn, will not look like thirdborn, and everybody will look alike. The reason why there's combination. Now, the S-shaped structure are called chiasmata. Chiasmata. Now, this place where the crossing over occur is called what? chiasmata. Thank you very much. Just take note of all those terms, please. We go to the next one, diakinesis. Let's come to the diagram also. Nuclear membrane fragmentation. Nuclear membrane is disappearing already. Everything disappears at the prophase. So then you don't see it again. Now, you will still have this crossing over here. Genetic material is still being exchanged between the two of them. It is the final stage, that's diakinesis. It is marked by terminalization of chiasmata. Chiasmata, if you remember, that is where crossing over occur. So it's going to terminate at this very stage. During this phase, the chromosomes are fully condensed. They are fully condensed, and the mitotic spindle is assembled to prepare the homologous chromosome for separation. Remember, when we are talking about metaphase in mitosis, we talk more of the spindle fibers. Are you getting me? So, which we said they basically help in migration, separation of what? Chromosomes. So to move from one place to another. So here also we are mentioning it because now we'll be going into what? The metaphase. So they started coming up. Meiotic spindle is assembled to prepare the homologous chromosome for what? Separation. By the end of the akinesis, the nucleolus disappears and the nuclear envelope also break down. The same thing happen in prophase of mitosis. It is at the prophase that the nucleolus disappear, the nuclear envelope also what? Break down. So diakinesis represents transition to metaphase. So diakinesis is more like the early stage of what? Of metaphase, because from diakinesis they go into metaphase. Now in metaphase 1 now, the bivalent chromosome are aligned on the equatorial plate. Now, from here, now it's more like what happened in metaphase, except for some little differences. Now, but definitely the bivalent chromosome. Remember now, the chromosomes are bivalent, even though they've separated. Are you getting me? But there's still some kind of crossing over. If you remember that, chiasmata. Aha. So the bivalent chromosome align. They now align as if they are still, since they are what's it called? They are homologous chromosome. So they align together at the same part. If you look at the diagram here, you see the alignment. You see the alignment. So the microtubules from the opposite poles of the spindle attached to the pair of the homologous chromosome. So as you can see here, so spindle fiber is coming, which is from microtubules. The spindle begin to form and begin to attach to them. Okay, sorry. And begin to attach so that it can draw them to where they need to be drawn to. That is at the metaphase. We are talking about the metaphase now. So they will not align, so the next thing can happen. So here now we have the anaphase and the telophase. So let's just continue, but I think you are aware of the whole process. So at the anaphase now, the homologous chromosome will now separate, while the sister chromatids remain associated at the what? The centromere. Homologous chromosome will now separate now. So the sister chromatids, remember, sister chromatids are two chromatids on the same chromosome. Non-sister chromatids are two chromatids, one from one homologous chromosome, another one from another homologous chromosome. So that is that. So the telophase now. Remember, homologous chromosome are separated here. Are you getting me? And when they separate, what are they trying to do? They begin to move towards the what? Towards each pole. If you remember the anaphase, the process is the same. Just some little adjustment, like I told you. At the telophase now, the nuclear membrane and the nucleolus reappears. Remember at the telophase, that is the stage where you now have the The chromosomes have now separated. So some move to one pole, the other one move to the other pole. Are you getting me? Then you begin to have the what? The so which means the nuclear the what's it called? Um um karyokinesis. It means karyokinesis has occurred. Division of the nucleus has occurred. Then followed by what? Cytokinesis, which is what? Cell the cytoplasm now dividing. So that is just the whole process here. So the nuclear membrane and nucleolus reappears because new one is being formed now. Cytokinesis follows, and this is called dyad of cells. So all those terms, I just want you to be familiar with them so that when you come across them, you will know what they are. Are you getting me? Now, what makes it different from mitosis here is because here now we are talking about homologous chromosomes still holding each other. But at the other one, just one chromosome, not homologous, no pairing, no crossing over, nothing in mitosis. It doesn't happen. But it happened here in meiosis. So, like I said again, the nuclear membrane and nuclear membrane that surrounds the nucleolus and the that surrounds the whole nuclear particles. It's called the nuclear membrane, which dissolved earlier. Now, now the at telophase they begin to what reappear, and nucleolus also reappear. So cytokinesis is then follows, and this is called dyad of cells. The same process as mitosis, just the little differences because here now there's homologous chromosome, there's what's it called? There's crossing over, there's chiasma, so it makes it different because of that little stuff. So the stage between the two meiotic divisions is called interkinesis. If you remember, interkinesis is more like interface. So instead of saying interface, so we call it what? Interkinesis. Because at that very stage, you will have production, synthesis of new cells to go ahead with another division. So interkinesis, and it is generally short-lived. Interkinesis is followed by prophase 2, a much simpler prophase than prophase 1. Now we've moved to meiosis 2 now. Remember, everything I explained is meiosis 1, and I said meiosis 1 is a bit longer than meiosis 2 because at meiosis 1, that is where you have crossing over, chiasma, homologous chromosome, tetrad, and all those stuff, chiasma or chiasmata. That is where you have it. So it takes a long process before all those things are formed, before crossing over occur, before there will be full exchange of genetic materials and everything. So it takes a long time. But at prophase meiosis 2 now, it's very, very short and more like that of prophase in mitosis. Look alike. Now, initiated immediately after cytokinesis, usually before the chromosomes have fully elongated. If you remember prophase, that is what we're talking about, still prophase. So in contrast to meiosis 1, meiosis 2 resembles the normal mitosis. That is just it. It looks like the normal mitosis. So the nuclear membrane disappears by the end of prophase 2. The chromosome again become compact. It becomes compact, which we've talked about before. The same, that's how it will be. No. Now, metaphase 2. Take it. Oh yeah, go. In metaphase 2, at this stage, the chromosomes are aligned at the equator, the same thing, and the microtubules from opposite side of the spindle get attached to the kinetochore of the sister chromatids. If you had gone through your mitosis, you will understand that process is the same thing, and that is just what you are seeing here. And then you will have this. Remember that at the end of meiosis 1, you already have two daughter cells produced. Now, each of the daughter cells now will now undergo the second meiosis, which will now form 1, 2, 3, and 4. And that's what I said from the beginning, that here now you have 4 daughter cells being produced instead of 2 daughter cells being produced under mitosis. So anaphase 2 now, it begins with the simultaneous splitting of the centromere of each chromosome, which was holding the sister chromatids together, allowing them to move to the opposite poles, up and down. So like what we have in mitosis. And at telophase now, meiosis ends with telophase 2, in which the two groups of central chromosome once again get closed by what? The nuclear envelope. So the cycle comes back again. So cytokinesis follows, resulting in the formation of tetrad of cells, 4 haploid daughter cells. 4 haploid daughter cells. So that explains everything under meiosis. So what are the significance of meiosis? Now, it helps in conservation of specific chromosome number of each species achieved across generation in sexual reproduction, reproducing organism, even though the process paradoxically results in reduction of chromosome number by what? By half. Now, what are we trying to say there? There is always a kind of conservation of specific chromosomes. Are you getting me? Now, so specific chromosomes will be conserved. In sperm cell, for example, if you look at sperm cell, we have millions of sperm cells. And if you look at them, inside each of the sperm cells, inside each of the sperm cells, inside each of the sperm cells, you have what we call different characteristics that is enclosed inside them. So from generation to generation, it is being enclosed and transferred to every new organism. So this continues. So it helps you to conserve the specific chromosomes. And also during crossing over, you will see variation. So that's the last one here that we have. Okay, second one here. It said it also increases the genetic variability. There will be variation in genetic constituents or genetic material. The reason why I will be different from my younger brother, from my younger sister, every one of us cannot look alike because there is what? Genetic variability in the population of organisms from one generation to the next. And lastly, it's variation are very important for the process of what? Evolution. Now, because there has been genetic material of variability, it leads to what? Variation, which is very important in the. So, differences between meiosis and mitosis. Now, meiosis, the production of offspring by sexual reproduction, including the fusion of gametes. Yeah, sexual reproduction, and there is fusion of what? Of gametes. So that is meiosis for you. Anything that has to do with sexual reproduction, meiosis. Gametes too being formed, meiosis. Now, in mitosis, it is an equational division, usually restricted to the diploid cells only. So that is why we talk about groups, because it's a diploid cell, which is the same as the somatic cell, the body cell, which undergo continuous division to produce new offspring. So gametes are formed from specialized diploid cells. So that's what I said. The diploid cells, that's the body cell, the somatic cell, are the ones that the meiosis make use of to break down the diploid cells into the gamete which is being needed. Okay, sorry for that. And next is it is concerned with reproduction, mainly sexual. So I think I've talked about that. Mainly sexual, it's basically for reproduction. That's for meiosis. And for mitosis, when you talk about growth, repair of worn out tissues, that's what mitosis is for. It takes place only in the production of gametes, meiosis. It takes place during growth of body or somatic cell. So the number of chromosomes of new cell is half the number in the parent cell. Yeah, so half of it, that's meiosis. For mitosis, it will be the same number of chromosomes that you see in offspring, also you see in parent. Four haploid cells are produced, four cells produced for haploid. And under mitosis, it is just two that is being produced, two offspring. Crossing over occurs in meiosis. Yes, there's crossing over in meiosis. There's nothing like crossing over in mitosis. So chromosomes are arranged in pair in a parent cell, but new cells have only one of each. So in parents, you have it paired, which homologous chromosome, we talk about that. But in the offspring, no, they are each of them stand on their own. Chromosomes are arranged in pair in both parents and the new offspring. That is under mitosis. Because the same way you have it in the parent is the same way you have it in the offspring under mitosis. Two O chromosomes form bivalent or tetrad. If you remember that, we mentioned that. So, but here now, there is no formation of bivalence under mitosis. Under meiosis, it involves only two stages of division. It will involve two stages of division under meiosis, but under mitosis, it is just one stage. And lastly, these are some of the questions that have been asked before under this very topic. So I want you to take note of them and go through them. That's why I said you should have your jotting pads and everything. You write it out the way you want to answer your question if they ask it. Very good one. Now they said in a tabular form, compare meiosis and mitosis with illustrated examples, meaning differentiate between mitosis and what? And meiosis. Sorry, compare between them. What are the similarities? What are the differences between them? So you give it to us. So similarities, differences in the tabular form. Yes, that is it. And B, outline the significance of mitosis. Mitosis, which we've talked about it earlier. What are the significance of what? Of mitosis. Importance of mitosis. You put it there too. Write short note on any four of the following. Now, those are under the, I think, number 2, basically in IGMB and in JPEB, which is the theory part. Short note on the following: mitosis, meiosis, matter, crossing over. So write short note on the following. So that's why as you are reading it, you need to understand it and jot things down. So in a tabular form also, compare mitosis and meiosis 1. You remember that? Aha, because I told you, I said mitosis and meiosis 2 look alike. They have almost the same thing. But mitosis and meiosis 1, they are different. So these are some of the questions you should be expecting from this very topic, which is cell division, mitosis and meiosis. So I want you to take time, answer all those questions, write it out. Yeah, during my own time when I was doing the same program, so what I do basically is for every topic being treated, I go to the past question, and thank God during your own time you have them being outlined here. I go to the past question, but here stop here. I think the question I have here is 2014, so this is 2020, take it 2024. So I will just advise you go into your past question, make sure you get your past question, go into your past question and begin to write out all the questions on this very particular topic and answer it the way you will answer it in the examination, so that every now and then you revise it. So when you come across it in the examination, you know what to write. So that is that about that. Thank you very much for your time and for your patience. So please let's try as much as possible to really go through all the things, and let's make sure we solve the questions on them. It's really going to help you. Really going to help you. These are the tactics I use during my own time. You read a whole topic, or the way you listen to it, and you will jot it out and answer the questions the same way you will answer it when you get to the examination or if you see the question. So thank you very much. Remain blessed. The next topic will be coming. That will be sale, and we'll be able to do more on that. Thank you very much, and remain blessed. Thank you.